Alright, so we're looking at the resting membrane potential of a cell. So when we look at a cell, we typically say that cells have a negative charge on the inside. And for every negative charge, there is a positive charge attracted to it on the outside of the cell. How do we get there? How does that even work? And it has to do with the concentrations of sodium and potassium. So inside the cell and outside the cell, we have different concentrations of these ions. Inside the cell, we only have about 10 millimoles of sodium, while outside we have about 140, and with potassium inside we have about 110, and on the outside we only have about 5. And so this is a big difference here, and there's a lot of energy that wants to move back and forth. And we can do this through channels. And now typically when we talk about sodium and potassium, we look at the sodium-potassium pump. But the thing is about that pump is that it only really accounts for the first 5 millivolts of our sodium, and the first 5 millivolts of our charge created by the sodium and potassium. But that is an important channel we have in here. And we also have voltage-gated sodium channels. And we also have potassium leak channels. And all 3 of these are going to be able to change the membrane potential. And so our sodium-potassium channels, we have 3 sodium out and 2 potassiums in. That kind of helps to first establish this gradient. Every now and then we have a potassium leak out. But for the most part, we keep our concentrations pretty regular. And when we look at a cell, the resting membrane potential is at about 70 millivolts. So we are at 70 millivolts. We're doing good. We're just kind of hanging out here. And how we find out that it's 70 is through, there's a bigger equation for it, but it gets its basis from something called the Nernst equation. The Nernst equation is the energy of the membrane equals negative 61 over Z times the log of the concentration of ions in over the concentrations of ions out. And so if we plug this in for potassium, we get about negative 90 millivolts. And if we plug this in for sodium, we get about positive 69 millivolts. And so our cellular energy isn't either of those, it's negative 70. So how do we get there? And so potassium is our primary driving force, or I will use that word better later. Potassium is our main player here. And so that's going to establish the majority of the gradient, but because of these potassium leak channels, it's not at that ideal potassium voltage. And so because that number is higher for sodium, it's going to drive our resting potential up a little bit, which is why we mainly rest at negative 70. And so if we look at the bigger equation, which is similar to the Nernst equation that we have down here, and it accounts for all of the ions in our system instead of just one, and that will give us something called membrane voltage, or VM. And also a correction to be made here. The resting amount of potassium is closer to 150 millimoles. These are all millimoles. Okay, so we have something called membrane voltage that comes from pretty much adding up all of the averages from the Nernst equations of all of the different ions we're looking at, so sodium, potassium, calcium, and chloride for the most part. And so, if we have our energy of whatever our ion is minus our membrane voltage, that is going to give us something called our driving force. And our driving force is essentially a quantification of how badly that ion wants to move in or out of the cell. And for sodium has a very high driving force to come inside of the cell, and potassium has a very high driving force to come out of the cell. Just because of their chemical gradients, first of all, is primarily what's making that strong driving force, but because of our electrical gradient, all of these negative charges, those potassiums want to stay put because these positively charged sodiums are attracted to these negative charges that have accumulated. And so, even though there are plenty of leak channels, they don't necessarily want to go anywhere very much. And so what happens when we play around with these concentrations, and what happens when we have a stimulus? So let's have a look here. Let's draw another little chart. Okay. So this will be 0, this will be negative 55 millivolts, and this will be negative 70 millivolts. And up here we have about positive 30 millivolts. So our cell is just going about our business until there's some sort of stimulus, and that stimulus causes some sodium channels to open. So now we have some amount of sodium coming into the cell for whatever reason, like there was a change in concentration, or maybe the cell next to us started letting sodium in. And so we will have what is called a graded potential right here. And this graded potential is pretty much just whenever the cell's resting membrane potential increases. And eventually what happens through that graded potential is we hit this negative 55 millivolts, and we spike. And we spike up to 30 millivolts. And what is happening here is those little sodium channels, those voltage-gated sodium channels, is all of these will pop open at about that negative 50 millivolts. Is there's enough positive force that pulls on this little spring, and it'll pull down, which pops this lid open, and sodium can rush inside the cell. And so that's why we have this spike here that goes up to 30 millivolts. And that's what causes this spike. And after a while, we start to open some of our potassium channels as well. Those will open up, so now we can have potassium moving out of the cell like it wants to. And that just drives the inside of our membranes voltage to just go up into the positives. But after a few milliseconds even, this little ball right here on our sodium channel is going to pop into the hole that was opened up. And so I'll draw it up here a little cleaner. This is open, this is our spring that's compressed down. This is our little ball that plugs up the hole. So our sodium can't come into the cell anymore. And now we start having our sodium-potassium pump start to pump some of that sodium out and bring some of that potassium back in. And so that's not going to do everything, but it's going to start bringing this down, and our channels start to close, but our potassium channels take a hot second to close, so it's going to come all the way down here before it goes back to 70. And so this little zone right here is what is referred to as hyperpolarization. And so hyperpolarization is because our potassium channels take long to close. They don't like to close very quickly. We get an influx of potassium more than the cell really needs, and down here we have a negative 90 millivolts. So like I was talking about earlier with that Nernst equation, and that potassium wants to have that difference of 90 millivolts, it's going to come down here before those weak channels can catch up and reestablish the kind of equilibrium concentration of sodium and potassium. And this is what is referred to as an action potential, is this red area up here. This is an action potential. And the difference between that and a graded potential is that a graded potential is just an increase of the cell's voltage, where an action potential is the sudden spike of sodium influx. And during this time, this is what is referred to as an absolute refractory period, where if there's another stimulus coming in, the cell is not going to be able to respond to that. And it's going to be here during our kind of reset phase, just because our potassium channels are slow and they don't want to respond again necessarily. And so this is going to be our absolute refractory phase, where no matter how great the stimulus is, the neuron or our cell is resetting and it's not going to accept any more input. But during this beginning phase, we have a relative refractory period, where it is possible to receive and process another stimulus if it's strong enough. And so if we suddenly get a stronger stimulus while we're right here per se, we can speed that up and spike it a lot sooner if we need to. And that's the relative refractory phase. Okay, and so when we're looking at neurons specifically, which I have some lovely clip art over here of, is when we're looking at neurons and how they like to communicate with each other is, here we have we'll start down here, is right here, this area that I'm going to do in yellow is called the axon hillock. And in order to send a message down this neuron to the other neuron, is that we have to get enough stimulus to cause an action potential right here in this yellow area. And so these branches, these dendrites, will be receiving stimulus. Action potentials will be firing down from here, from all these different places, from all the different synapses it has. And it will have this positive charge. Every different stimulus will have a positive charge that it's receiving. And it will send these through an electric field. It will communicate this positivity down the dendrites of the neuron. And we hope that it can get here and fire off an action potential. And it can go down to the next neuron. And sometimes these can be referred to as excitatory or inhibitive, because sometimes there might be like a synapse here that activates something that might cancel out one of these, and so you wouldn't get as strong as a signal coming down this axon. And so we have something what is referred to as the length constant, which is shown as lambda. And so lambda equals the square root of Rm over Ri. Ri is internal resistance, which exists in all kind of electrical systems. Yes, it exists in pretty much all electrical systems, and it is the natural resistance of whatever material you're trying to send an electrical signal down. And in neurons, because we're trying to send down this positive charge, there will be like negatively charged proteins in here that will absorb some of that positive electric field we're trying to move, and it can cause problems with being able to send those things. And so one way we can decrease Ri is to make our neurons bigger. Cannot make bigger. And so cephalopods, mainly squids and octopuses, love to do this. They love to make big, thick, meaty neurons that have very little internal resistance. Just because if we make this neuron like double the size, and we just have this little protein here, then we're still going to have plenty of space to get that positive charge to travel down. Now there's also Rm, which is membrane resistance. And so with our membrane, we have stuff moving in and out all of the time. And those ions and those things coming in and those things going out can also get in the way of that positive electric field we're trying to send. And so if our membrane has a very high resistance to letting things in and out, it doesn't like to do that very easily, it's also going to allow for a longer length constant, and we're going to be able to get that positive electric field to move further. And one way that mammals love to do this, especially in our big neurons, is we will produce these sheets of lipid cells called Schwann cells, and these are called myelin. And it's this thick layer of lipids that doesn't let in pretty much any positive charge that will be accumulating on the outsides of these cells. And so since there's like no really any charge moving in here, then we're going to be able to let that positive signal go down the axon very quickly. And at these little spaces between the myelin sheets, we have what are called the nodes of Ranvier. And this is where we're going to have our voltage-gated channels. It's where we're going to have all of our membrane channels. So we have a little voltage-gated sodium channel, here we have a potassium leak channel, all sorts of things is where we're going to have all of our different kinds of channels and moving things in our nodes of Ranvier. And so, since this positive signal is coming down this axon really quickly, it's going to be able to trigger this gated channel very easily, and the influx of sodium right here through this channel is going to make another strong positive signal to travel down the axon. And so if we have long axons with lots of myelin and the nodes in between them, they can go pop, 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 and run down very quickly. And it's very good to have in our neurons, just so we can do things efficiently in our bodies. Until we get down to our synapses here. So there's lots of vesicles in our synapses, mostly filled with things like glutamate, sometimes acetylcholine if we're looking at more like muscle neurons, but in the brain we typically use glutamate. And on these vesicles we have something called snare proteins that are these little twist tie thingies, and we also have a bunch of twist tie snare proteins on the walls or on the membrane of our neurons as well. So that when this positive charge comes rushing down, it's going to activate these snare proteins, and these vesicles will travel to the membrane, connect with the snare proteins there, and twist and twist and twist together until that vesicle fuses with the membrane and pops and releases its contents into the synapse space. And then that synapse space will have like ligand receptors that will open sodium channels, so we can start this process all over again and send down more positive signals. And another factor of getting those signals across quickly is capacitance, because our neurons can work like capacitors. So we have a big, kind of thin, stretchy membrane right here. In this place, we have a larger distance. And so we have some positive charges here, some negative charges here. Here we have a lot of positive charges and a lot of negative charges. And so in this system where, let's say we have some proteins in this membrane, so these positive charges are really close together, they're really packed, they're going to be wanting to repel each other. They are not enjoying being so tightly packed. And they can interact with these proteins here and send down a positive force. And these negative charges down here, they aren't super happy to be packed together either. They're also going to be interacting with these proteins. And what the proteins help to do is it helps the positive and the negative charges to quote-unquote feel each other through this membrane space. And if they can feel each other, that attractive force will eventually overcome the repulsion force of the like charges next to it. But in this system, where there's not a lot of charges and there's a decent amount of space between them, these are going to be actually pulling harder on each other, because that's the main force we have in play here. Like, these are separated far enough apart that they're pretty much just going to mind their own business, because our attraction force, just as is a lot stronger than our repulsive force. So this is a system with high capacitance, and this is a system with low capacitance. And where this comes into play is in our myelin, because where we have these few positive charges on the outside and the negative charges inside our cell, is these are so kind of spread far out that these are just going to be busy trying to interact with each other, that they won't super interfere all that much like in this high capacitance system. And so this low capacitance is going to make our signals travel faster down our myelinated neurons.